For 20 years, one number has defined everything the modern world thinks it knows about the genetic legacy of the Mongol conquests. 1 in 200 men on Earth today, according to that number, is a direct patrilineal descendant of Genghis Khan. Roughly 16 million living men, spread across a vast arc from China to Eastern Europe, carrying in their Y-chromosomes the genetic signature of the most prolific conqueror in recorded history. The number appeared in a 2003 paper in the American Journal of Human Genetics, spread through textbooks and documentaries and dinner table conversations worldwide, and became one of the most repeated statistics in the history of popular science. It transformed Genghis Khan from a historical figure into a genetic phenomenon, the ultimate patriarch, the man whose biological reach across the centuries was as vast as the empire his armies had carved across 2 continents. Then, in February 2026, a paper published in the Proceedings of the National Academy of Sciences did something that the 2003 study could never have done. It extracted DNA from the actual graves of the men who ruled in Genghis Khan's name, and what it found inside those graves does not match what the world has been told for 2 decades. The 2003 study that launched the 16 million claim was a genuine scientific achievement built on the tools available at the time. An international team analyzed Y-chromosome data from more than 40 modern populations across the former extent of the Mongol Empire. They identified a specific cluster of Y-chromosome variants, the haplogroup designated C3, also known as the C-star cluster, that appeared at unusually high frequencies across a vast swath of Asia from China to Central Asia. The distribution pattern was consistent with rapid expansion from a single ancestor, approximately 1,000 years ago, precisely when the Mongol Empire was at its peak. The timing and geography pointed toward Genghis Khan. The researchers concluded that conclusion rested on an important assumption, that the C3e lineage found at high frequencies across modern Asian populations was the same lineage that Genghis Khan and his ruling family actually carried. The 2026 PNAS study tested that assumption directly, and the assumption did not hold. Like and subscribe, because what the ancient DNA of the Golden Horde's actual ruling elite showed is the finding that changes the entire picture of the Mongol genetic legacy. The 2026 research, published in PNAS in February and led by Aiken Askapouli of the University of Wisconsin-Madison, extracted and analyzed genomes from 4 medieval individuals buried in elite mausoleums in the Ulitau region of central Kazakhstan. The graves belonged to members of the Golden Horde, the northwestern branch of the Mongol Empire founded by descendants of Genghis Khan. The Ulitau region was at the geographic heart of the Golden Horde's core territory, and local Kazakh tradition has long held that one of the 4 mausoleums studied contains the remains of Jochi himself, Genghis Khan's eldest son and the founder of the Golden Horde dynasty. The Golden Horde elites primarily derived their genomes from ancient Northeast Asians, with an additional ancestral component from either ancient North Eurasians or a bear, a Scythian-related population, such as the Kipchaks. That ancestral profile is precisely what you would expect from the ruling class of a Mongolian steppe empire, a predominantly East Asian genetic foundation with admixture from the steppe and Central Asian populations the Mongols encountered and absorbed during their conquests. The critical finding concerns the Y-chromosome. By analyzing individuals from medieval elite mausoleums in central Kazakhstan, researchers confirmed the association between the Y-chromosome haplogroup C3 Tos and the Mongol Empire. The 3 men in the mausoleums did carry C3 guis, the haplogroup long associated with Genghis Khan's dynasty. That confirmation was itself important, providing the first direct ancient DNA evidence that real documented Mongol ruling elites carried this haplogroup rather than it, being an inference from modern population distributions. But, and this is the finding that rewrites the standard narrative, these elites possessed a specific, remarkably rare sub-branch of C3 wax, not the dominant version found in millions of men across Eurasia today. The sub-branch carried by actual Golden Horde ruling elites is not the same sub-branch that shows up at high frequencies in modern Asian populations, and that was used to calculate the 16 million figure. The data suggest genetic signal tracks with the empire's expansion, but likely reflects contributions from multiple influential lineages, rather than a single patriarch. Like and subscribe, because what this sub-branch distinction actually means for the 16 million claim is something no mainstream media coverage of this paper has fully explained. The C3-Siksu haplogroup is a broad category. Within it, multiple distinct branches diverged from each other at different points in history. When the 2003 study identified a widespread C3-2 cluster in modern Asian populations, it was working with the resolution available in 2003, which was sufficient to identify the broad haplogroup but not to distinguish between its sub-branches with the precision that modern genomic sequencing provides. The widespread C3 lineage that appears in roughly 0.5% of men worldwide The lineage behind the 16 million claim belongs to one sub-branch. The C3 lineage carried by the actual Golden Horde ruling elites in the 2026 study belongs to a different sub-branch, one that is considerably rarer in modern populations. The findings suggest the widespread belief that 1 in 200 men descends from Genghis Khan may be overstated, as the specific genetic lineage remains uncertain without his confirmed remains. This does not mean the 16 million claim is simply wrong in every sense. The widespread modern C3 sub-branch did expand dramatically during the period of Mongol dominance, and its distribution is almost certainly connected to the Mongol conquests in some way. But the connection may not be what the original 2003 study implied. The widespread geographic distribution of C3 is indeed a legacy of the Mongols, but not necessarily because Genghis and his heirs specifically had so many offspring. It may instead be because ordinary Mongol peoples sharing the same Y-chromosome heritage shared more generally in the conquests and dispersal led by Genghis and his heirs. In other words, the 16 million men may be descendants not of Genghis Khan himself, but of the broader Mongolian population, whose members participated in the conquests, settled in conquered territories, and reproduced across the empire's vast territory. That is still an extraordinary genetic legacy of the Mongol expansion. But it is a fundamentally different claim than the one that made Genghis Khan the ultimate patriarch in popular imagination. The claim that one specific man's Y-chromosome is walking around in 16 million living bodies today. The 2026 study also addressed one of the most contested questions in the entire history of Mongol scholarship, the paternity of Jochi. Genghis Khan's eldest son was born shortly after his mother, Berta, was held captive by an enemy tribe, and doubts about whether Jochi was Genghis Khan's biological son circulated during his own lifetime and have never been definitively resolved by the historical record. Although the local traditions about the occupants of the mausoleums were not supported by the DNA results, traditional stories relating specific tombs to specific individuals like Jochi were not confirmed. The genetic evidence places the buried elites within a clear Mongolian lineage genetically connected to the Mongolian plateau. The Y-chromosome lineage found in the Golden Horde mausoleums is linked through identity by descent analysis to ancient genomes from Mongolia itself, establishing a direct genetic connection between the Golden Horde ruling class and the Mongolian heartland. That connection is consistent with the historical claim that Jochi was Genghis Khan's son, because a ruling class that was not paternally connected to the Mongolian plateau would show a different genetic signature. The data does not prove the paternity claim, but it is more consistent with Jochi being Genghis Khan's biological son than with him being someone else's. Like and subscribe, because the broader story of what ancient DNA has revealed about the Mongol conquest's genetic impact on the populations it swept over is even more consequential than the question of Genghis Khan's personal descendants. The Max Planck Institute study of ancient Mongolian genetics, which analyzed 214 ancient individuals spanning 6,000 years of Mongolian plateau population history, revealed something remarkable about what happened genetically during the Mongol Empire period itself. 1. Individuals from the Mongol Empire showed a marked increase in Eastern Eurasian ancestry compared to individuals from the earlier Xiongnu, Turkic, and Uyghur periods, accompanied by a near-complete loss of the ancient ANE ancestry that had been present since before the Xiongnu Empire. By the end of the Mongol Empire, the genetic makeup of the eastern steppe had changed dramatically, ultimately stabilizing into the genetic profile observed among present-day Mongolians. That dramatic genetic shift during the Mongol Empire period, within Mongolia itself, not in the conquered territories, suggests that the political and military success of the Mongol expansion was accompanied by significant demographic change on the Mongolian plateau itself. The populations that participated in the conquest were themselves transformed by the processes of empire building, migration, and population movement that conquest set in motion. What the genetic record of the conquered territories shows is equally significant, and equally at odds with the simplified narrative of Mongol demographic dominance. The Mongol Empire, at its peak in the mid-13th century, controlled territory stretching from the Pacific coast of China to the Danube River, approximately 24 million square kilometers, roughly a quarter of the total land surface of Earth. That empire was governed by Mongol military elites, but administered through existing local bureaucratic structures and populated predominantly by the conquered peoples, Chinese, Persians, Russians, Poles, Koreans, Arabs, and dozens of other populations that vastly outnumbered the Mongol conquerors in every territory they controlled. The genetic evidence from conquered populations consistently shows that Mongol rule, for all its military violence and demographic disruption, did not produce the wholesale genetic replacement of the populations it conquered. The administrative and military elites were Mongolian. The genetic substrate of the populations they governed remained predominantly local. This finding matters because it challenges the most extreme version of the conquest narrative. The image of Mongol armies sweeping across continents and not merely defeating, but genetically replacing the peoples they encountered. The evidence consistently points toward a different model, one of Mongol ruling elites maintaining genetic distinctiveness above a largely undisturbed local population for the duration of the empire, followed by rapid genetic assimilation as the empire fragmented, and Mongol ruling families blended with the local gene pools of the territories they governed. Archaeological evidence sheds light on a medieval population undergoing religious and cultural transition, offering insights into the societal changes experienced by Mongolian conquerors. The Golden Horde mausoleums themselves tell that story materially. The burial practices visible in the Yuletau sites show a ruling class navigating between Mongolian steppe traditions and the Islamic culture of the Central Asian populations they governed, reflecting a process of cultural integration that the genetic data confirms was matched by biological mixing between the Mongolian elite and local Kipchak and Central Asian populations. Like and subscribe, because the question of Genghis Khan's own Y-chromosome, the one piece of evidence that would definitively answer what his actual genetic legacy was, remains the most famous unsolved problem in ancient DNA research. His burial site has never been confirmed. According to historical tradition, the funeral procession killed everyone who witnessed it, and the grave was deliberately concealed, with accounts suggesting that horses were driven over it repeatedly to eliminate any trace. The Khenti Mountains of northeastern Mongolia, near his birthplace, have been searched by multiple expeditions using ground-penetrating radar, LiDAR, surveys, and traditional archaeological methods without producing a confirmed identification. Without verified remains from the conqueror or an unquestionably identified close male descendant, the most famous family tree in Eurasian history remains genetically unfinished. The 2026 PNAS study has moved the search substantially forward. By confirming the specific C3 sub-branch associated with actual Golden Horde ruling elites, it has narrowed the genetic profile that Genghis Khan's own remains, if found, should match. But the tomb itself has not been located, and without it, the definitive answer cannot be written. What the accumulating body of ancient DNA research has produced, the 2026 PNAS study, the Max Planck Mongolia dataset, the broader genetic history of populations across the former Mongol Empire, is a picture of the Mongol conquest story that is simultaneously smaller and larger than the popular version. Smaller because the specific claim that one man's Y chromosome lives in 16 million bodies today is almost certainly overstated, with the actual Genghisid Y chromosome subbranch being considerably rarer than the widespread modern lineage it was confused with. Larger because the demographic and genetic transformation that the Mongol conquests set in motion across Eurasia, the accelerated mixing of populations across thousands of kilometers of steppe and beyond, the genetic shifts within Mongolia itself, the cultural and biological integration of Mongolian elites with the peoples they governed, was a genuine continental scale demographic event, even if it did not operate through the simple mechanism of one man's extraordinary reproductive success. The scientists who built the 16 million claim were not wrong that something remarkable happened genetically during the Mongol period. They were working with the resolution available to them in 2003. What they could not know was that the widespread C3 lineage they were tracking was not quite the lineage they thought it was, and that the actual Y chromosome of the Mongolian ruling elite was rarer, more specific, and more precisely connected to the Mongolian plateau than the story that made Genghis Khan a genetic phenomenon across the entire modern world. The question of how the Mongol genetic footprint is distributed across modern populations, and which specific populations carry the clearest signal of the empire's demographic impact, is one that the 2026 findings have substantially refined. Before the PNAS paper, researchers working from modern population data had identified high frequencies of C3-related lineages in populations across Central Asia, Siberia, parts of China, Mongolia, and extending into parts of Eastern Europe. The geographic extent of that distribution was taken as evidence for the reach of Genghis Khan's specific genetic contribution. After the 2026 paper's clarification that the widespread modern C3T lineage and the elite Mongolian ruling lineage belong to different subbranches, the interpretation of that modern distribution needs to be revised. The populations carrying the widespread modern C3T lineage are almost certainly carrying something genetically connected to the Mongol expansion, but they may be carrying the genetic legacy of ordinary Mongolian soldiers, administrators, and settlers, rather than the specific patrilineal descent from Genghis Khan that the original claim implied. That distinction matters for understanding what kind of demographic event the Mongol Empire actually was. The traditional narrative, amplified by the 16 million figure, presented the Mongol conquests as primarily a story of elite reproductive success, with Genghis Khan and his sons personally generating an extraordinary number of descendants through the preferential access to women that conquest afforded. That narrative is not entirely wrong. The historical sources are clear that Mongol rulers maintained large harems and produced numerous children, but the genetic evidence suggests the demographic impact of the conquest was distributed more broadly across the Mongolian population than a single patriarch model implied. It was an empire, and empires move populations. The soldiers who carried out the conquests, the administrators who governed conquered territories, the merchants who traveled Silk Road routes now pacified by Mongol power, the settlers who followed military campaigns into newly controlled lands, all of these people moved, mixed with local populations, and left genetic traces that the modern distribution of C3-related lineages reflects. The empire's genetic legacy is the legacy of that entire mobile population, and the legacy of a single man's Y-chromosome replicated through extraordinary personal reproduction. Like and subscribe, because the Xiongnu comparison illuminates the Mongol conquest story in a way that no amount of analysis of Mongol-era populations alone could produce. The Xiongnu Empire, which preceded the Mongols on the Mongolian plateau by roughly 500 years, offers a genetic template for understanding how steppe nomadic empires actually shaped population history. Ancient DNA study of the Xiongnu found that the empire was multi-ethnic, with high genetic diversity found across the empire and even within individual extended elite families. At the fringes of the empire, women held the highest positions of power, and the highest genetic diversity was found among low-status male servants. That pattern, diverse genetics at the margins and among the lower social strata, with somewhat more restricted diversity among the ruling class, is consistent with a model of steppe empire building, in which conquest incorporates diverse populations into a politically unified structure, while maintaining distinct social hierarchies that partly preserve genetic distinctions between the conquering and conquered populations. He, Mongol Empire, almost certainly operated on similar principles, with the genetic distinctiveness of the Mongolian ruling class maintained partly through preferential marriage within the Borjigin clan and other elite Mongolian families, while the broader Mongolian population that participated in the conquests mixed more freely with the diverse populations of the conquered territories. The Max Planck Mongolia studies finding that Mongol Empire individuals showed a dramatic shift toward Eastern Eurasian ancestry compared to the Xiongnu and Turkic periods provides an important baseline for understanding what made the Mongol demographic moment historically distinct. The Xiongnu had incorporated Western steppe ancestry into their population in a way that created significant genetic diversity across the empire. The Mongol Empire reversed that pattern in Mongolia itself, concentrating Eastern Eurasian ancestry as the dominant genetic signal in ways that stabilized into the genetic profile of modern Mongolians. Understanding why that inward genetic consolidation happened at the same time the empire was sending military forces to the farthest reaches of Eurasia requires understanding the specific political and demographic mechanisms of Mongol imperial expansion, mechanisms that ancient DNA research is only beginning to illuminate with the granularity needed to answer such questions precisely. What the ancient DNA evidence has collectively produced: the PANAS 2026 Elite Mausoleum Study, the Max Planck Mongolia. Genetic history, the broader population genetics of the former empire, is a conquest story that is more complex, more accurate, and ultimately more interesting than the version that circulated for 2 decades behind the banner of 16 million Genghis Khan descendants. The Mongol conquests were a genuine continental-scale demographic event. The Y-chromosome signature of the Mongolian ruling class is real and confirmed in actual elite burials for the first time. The genetic footprint of Mongolian populations across the former empire is visible and meaningful, but the simple narrative, one man, one chromosome, 16 million living descendants, has been replaced by a more precise and more scientifically defensible account in which the empire's genetic legacy is distributed across the Mongolian population that created it rather than concentrated in the personal reproductive success of its founder. Scientists were not wrong that the Mongols changed the genetic landscape of Eurasia. They were wrong about the mechanism. And the difference between those two things is the story that ancient DNA is now in a position to tell.